AgResearch and Grasslands Technology, along with PGG Wrightson and Seeds, are developing new pest-resistant, animal-safe endophytes for ryegrass. Because a type of gene editing technology is needed for this work, it's undertaken in secure containment facilities. Grasslands Technology CEO John Carradis explains.
Grasslands Technology is a wholly owned subsidiary of AgResearch, and our role is to invest in the development of technologies and products that can then be commercialised for use by pastoral agricultural farmers in New Zealand. A very important part of Grasslands Technology's product portfolio is the development of endophytes for ryegrass in New Zealand, which allows ryegrass to persist against insect pests in particular. We've been exploiting the natural variation in endophytes for over 20 years now, but we've got to a point really where we need to look at new ways of developing that variation. And so gene editing has become an option for us to look at. So we're in the glasshouse complex at AgResearch in Palmerston North. The glasshouse is a PC2 plus glasshouse, which means it's completely contained, and there is no water or air that leaves that building without being filtered and removing any living organisms or parts of living organisms from it. The research that we're talking about with regard to gene editing endophytes in ryegrass can only be done in New Zealand in containment. The earlier genetic modification tools used a somewhat more random way of inserting genes into the DNA of organisms. And that was done in a way that you then grew hundreds or thousands of the transformed organisms to find the one that expressed the trait appropriately. The difference with the gene editing that we're using today is that it's much more targeted, and it's guided to actually insert or delete the gene of interest into the DNA of the plant in the appropriate place. So we don't need to grow out the hundreds and thousands of plants to identify the trait we're looking for, because it's a lot more targeted and we understand a lot more about the gene it's actually affecting or inserting into the genome. That's excellent. Look forward to that.
An endophyte, they're actually a group of microorganisms, primarily fungi and bacteria. They live within plants, and the term endophyte actually means endo within and phyte plant. In nature, all plants contain endophyte microorganisms. And the plants that we're interested in, ryegrass and grasses in general, they contain a special group of endophytes that are really beneficial, and they protect the plants from various stresses. So they can be biotic from pests and diseases, or abiotic, which is from drought, extreme temperatures, and some endophytes as well even improve nutrient acquisition. So they're really amazing organisms, and we've harnessed them for sustainable agriculture in New Zealand to protect ryegrass. AgResearch has been looking and discovering these endophytes in ryegrass for over 40 years. But what we keep finding is that from our collection of endophytes, mostly they comprise endophytes that have chemicals or chemistry that causes animal toxicosis. So in New Zealand, most farmers will be aware of ryegrass staggers, which is caused from a compound that's tremogenic. And in the US, one of the big problems is fescue toxicosis, which is caused by another compound, which is a vasoconstrictor of the blood. So those are the sort of bad compounds. And what we find predominantly is that many endophytes that we either collect in actual collection trips around the world and bring that seed back and look for endophytes, as we find they often express these toxic substances. One of the reasons why we're interested in these ryegrass endophytes is because they provide pest resistance. And a couple of the important introduced pasture pests are Argentine stem weevil and African black beetle, plus there's a number of endemic insect pests as well. So what the endophyte does is it produces these compounds that deter those insects, and that means that our pastures have great persistence, which is obviously what we want. What we chose to do is use one of the newer gene editing technologies, CRISPR-Cas9. And so this system is a great tool. It's very precise, so it's enabled us to go in and target the gene of interest, which is responsible for expressing a compound that could be an insect deterrent compound or a toxic compound to animals, and we've actually been able to shut it off. So we've been able to do this in a way which is aligned to the SDN1 standard. So SDN1 means site-directed nuclease, and it's just a way of categorising the different types of edits that you can make in the lab. Now, the SDN1 is really important to understand because in many countries it's not regulated at all. And this is because the organism itself makes the repair. So essentially what happens is Cas9 is an enzyme and it comes and makes a double-stranded cut in the DNA. And the organism itself tries to repair that cut, but it makes a mistake and the organism itself creates the mutation. And that mutation will delete the gene function. And so we've been able to remove those toxic compounds that are being made, but still keep those good compounds being expressed. There's many tests that you can do in the lab, but what we're trying to achieve is a product for farmers, and we have to understand what happens in a field environment. And so what we're able to do here in New Zealand is quite limited because of our regulations. All we can do is grow our plants in our containment glasshouse, which is under negative pressure. But in a glasshouse environment, it's really hard to produce lots of seed. And so what we've had to do is produce from each of our edited lines small quantities of seed that have then been sent over to Australia where the GM regulations are different. And it's been sown and multiplied. Ultimately, New Zealand is the country that needs to benefit from this particular product because we need perennial ryegrass that performs well. And we want to make sure we are thinking about keeping our inputs low, that we have environmentally sustainable solutions. And the Endophyte provides that because you don't need synthetic pesticides when you have this Endophyte. And what we've done with gene editing is remove sort of the bad part of the Endophyte, which is the animal toxicosis, while just keeping the good part. So it's an excellent solution for farmers.